This research was actually a part of my PhD, and in this research we actually consider patients with Parkinson's disease undergoing deep brain stimulation, and with a double aim. On the first part, we actually wanted to investigate the impact on sleep, not only on sleep quality but also on sleep structure by means of video polysomnography of deep brain stimulation, to see how sleep architecture was modified by deep brain stimulation. And what we saw is that actually going along to a better sleep quality, this also was consequential to an increase of deep sleep and a small decrease of light sleep, even if circadian patterns didn't actually change. On the other hand, thanks to the fact that nowadays we have the possibility with new technologies to record what we are stimulating. We consider a patient with deep brain stimulation in the subthalamic nucleus, and therefore we were able to record the local field potential in the subthalamic nucleus during the night, synchronized with video polysomnography. So with not only the EEG during sleep, but with all polysomnographic signals. And this was important for several reasons. First, it is important to try to find signatures, intracranial signatures of sleep, because nowadays we have the possibility to adapt the stimulation with these devices depending on the recordings of the local field potentials. And this is done usually during the day if the patient finds in more active, more on, or more inactive of where the patient may have more motor problems. And this is nowadays done on specific bands, especially the beta bands. But we don't know specifically how this band behave during sleep. And it would be good also to inform these devices of what is happening in sleep, for example, by reducing the stimulation during sleep or adapting the stimulation when before the patient actually wakes up during arousals and subsequent awakening during the night. And another point was into delving into the neurophysiological patterns of REM sleep behavior disorder, because by recording it, this patient with Parkinson's disease also had REM sleep behavior disorder, and the hypothesis is that this patient moving better during REM sleep behavior disorder episodes, the basal ganglia may not be included into the REM sleep behavior disorder patterns. Some nuclear medicine evidence suggested that these nuclei do not activate during this part of the episode and in this disorder. We were able to record hence the subthalamic nucleus during REM sleep behavior disorder episode, and what we actually found, we found actually a change of the activation pattern of local field potential during this episode, also distinguishing simple motor activation during REM sleep with more complex episodes, suggesting that even if not primary, there is actually an activation of the basal ganglia during this type of episode in REM sleep behavior disorder episode. Therefore, I believe with this study we try to increase the knowledge by giving data to allow in the future a real sleep-aware deep brain stimulation, and also on the other side to delve more in the neurophysiology of these disorders.